Hi, Don.
Oh, I was mute. Good morning.
Good morning. How are you?
Pretty good.
Good. It looks like we're able to communicate. Do you want to try and share your screen?
Sure. It says host disabled attendee screen sharing.
That's totally weird. Let me see what I can do about that. Who can share? Do it that way. Who can start sharing when someone else is sharing? Try that and see if that allows you. I love this stuff.
I've got desktop one, I guess is me, right?
Yeah, do you just have one screen?
Yeah.
Yeah.
Open system preferences to grant access. Okay, hold on. That's me, I think.
I think so.
Oh, privacy. Screen recording. Oh, here it is, Zoom. Okay. Oh, you were desktop one. Whoops. So let me do this one. Share that one. Okay.
Okay, there we are. Awesome. And can your pointer move if you, yep.
Yeah.
All of that looks great.
Now let me do slideshow.
Yeah.
How do I hide that? Can you see across the slide my Zoom control bar?
No, I don't see
Okay,
it.
you just see the slide? Okay, good.
I think you're okay. I'm going to just go over a couple of people I need to resend the Zoom to because they're having a problem. So I'll visit back with you in just a sec. Let me see if I can get that taken care of.
Okay.
Let's see. Okay, hopefully that worked. How are you, Don?
I'm doing well. A little, I guess just like everybody else, a little bit ready for this to be over.
Yeah.
You know?
Yeah. It's hard to be patient, clearly.
Yeah.
Yeah, unfortunately.
But get along pretty well. So.
Good. Everybody's healthy?
Yeah.
Your family?
Yeah, doing well. One son called the other night and said, "Hey, Dad, I got married."
Oh, no kidding?
Yeah. We had known they'd been planning, but, you know, she's a Chinese national who works on quantum computing.
Yeah.
And wanted to go do her postdoc. She's graduating from Caltech and wanted to go do her postdoc in Canada. And they were just afraid, you know, if she goes up for an interview, she won't get back in again.
Oh.
And so they figured, let's just go on and do it. We'll have a party later.
Sure, sure. That makes sense.
Engaged a while, so
With the current administration, you never know.
Well, another son put his wedding off, so they could have a party because they have friends who would be coming from Texas and Florida, so.
Jeez, those are. Mine is simple. Everybody's in their locations, being safe, doing their thing. And I have one daughter who's a physical therapist who's actually on in Reno, Nevada. She's on the.
I don't know how to use Zoom. I tried to join in, but I don't know how to mute myself. Can I see your?
If you don't,
I leave the meeting.
How can I help in individuals muting?
Can I say something?
Sure.
This is Minu from Dr. Philpott's lab. Some of us did not get the link.
I just sent it again. Did you? I don't know. Are you on that group?
Let me check. I mean, if you don't see people that.
Hang on just a minute and let me just do this again. I see your email that went to Shelly. Let me just try this again. Okay, so I just sent it again, Minos, to you directly.
Yeah, Olga gave it to me. I'm just telling you, if you don't see people, that means they didn't get the link. Thank you.
Okay.
No, I didn't get email. Yes, I did.
Okay.
Thanks.
Yeah.
Thank you.
Of course. Glad to have you. We'll give just a couple more minutes before we go. And then hi, Tom.
How's it going?
Good. How are you doing?
Doing okay. Nice meeting. I really haven't been enjoying it.
Oh, I'm glad. Good.
I think you'll be on the BioIron schedule, correct?
Yes, I think we are. So that's wonderful. Hopefully we'll continue to be able to do some of these, but also be back in person at some point.
Oh, we'd love to. I just got a note from a friend in Italy that they're returning to pretty much normal.
Oh, I'm glad to hear that.
In Lombardy, mind you, at the epicenter of the pandemic, they're returning to normal. So there is hope.
And if we keep working those vaccines, maybe we'll all be able to start moving around.
Well, I think that's going to take a while. I think we have to get it under control before then. Yeah.
Well, I don't know about you guys. Utah's not doing a great job.
No, we're not. Los Angeles has the highest cases, highest number.
Geneva says. I hope that
I'm gonna.
Hi everyone, I can contribute from Milan, and I confirm
Wonderful.
that you are doing better,
Welcome,
relatively
welcome.
better. Yes.
Great. Nice to see all of you.
Thank you for putting up this webinar.
Hello.
Okay, great. I think we'll get started. My name is Diane Ward, and I'm the Enrichment Chair for the NIDDK-sponsored Centers for Iron and Heme Disorders at the University of Utah, and we've organized this virtual seminar series. And today it's my pleasure to introduce you to Don McClain. I became acquainted with Don as he was a faculty member here at the University of Utah. Don received his bachelor's degree from Haverford College. I'm going to mute everybody. Okay. Can you still hear me, everyone? Okay, thumbs up. Thank you. Okay. Don received his bachelor's degree from Haverford College and then went on to get his MD Ph.D. at Rockefeller University. He did his internship and residency at Stanford and a fellowship at UC San Diego in internal medicine. Don then became an expert in diabetes, essentially, and focused in on endocrinology and I don't even know, Don, if you first came to Utah or where did you land after?
Oh, I've been a couple of places for then, UCSD and UAB, and but I was in Utah longest I've ever been any place on the planet.
Okay. Okay, and he now resides at Wake Forest University, and he's going to tell us about diabetes and iron. Can't live with it, can't live without it. And we want to welcome you, Don, and welcome everybody who's participating. We have a chat area where you can enter questions in, and I will try and moderate those questions, some during the presentation if they seem appropriate to ask at the time, and some that we'll do at the end, if that seems okay with you, Don.
Yeah.
Okay, we will let you share the screen now.
Are you seeing it? It's shared, right?
Yes, it is shared.
Okay. It's a pleasure to be here today. This is our new med school here in Winston-Salem, built in an old cigarette factory donated by R.J. Reynolds, who's no longer in town, I guess, but well, their company is, but they don't make cigarettes here. So I'm gonna, oops, wrong one. There we go. This is just an outline, and I apologize. Some of this stuff, obviously, this group is going to know, but and some of it is work that has been published for some time, and I apologize for that. But I do see a lot of new names, so I will start kind of from ground zero. Just a brief evolutionary perspective on iron homeostasis, which makes me nervous in this august crowd to which I'm speaking. A little bit about pathologic iron overload in diabetes, the central role of the adipocyte, the fat cell, in integrating iron levels with metabolic regulation, and then a bit about normal dietary iron, that is the iron levels we all walk around with in diabetes, with some mention of other chronic diseases also related. And again, here you all know this, that 6% of the Earth's crust is iron, a potentially dangerous oxidant when reacting, just for one example, with peroxide to get to generate free radicals. So anyway, because of this, the theme, a theme in early evolution was to protect organisms from iron and bring it in only when needed. And this was true through early hominid evolution, when we were still eating meat and sedge grasses prior to the agrarian revolution. This potential danger of iron, though, coupled with its need for redox reactions, led to this coupling of glucose metabolism with iron. And it's well demonstrated in yeast, for example, that will not import iron into cells unless they need it for oxidation reactions. And if they can get enough energy from fermentation, they won't bring the iron in, and conversely, they can't convert to oxidative metabolism unless iron is available. And you're all familiar with this. This is the elaborate mechanism that's evolved in humans for the regulation of iron. And this regulation is really very well developed and expensive to the organism. Jerry Kaplan used to tell me that there were 50 genes at least devoted to chaperoning iron around the cell, and there are probably a very large number of iron chaperones that we're not even aware of yet in terms of their total function and moving iron within the cell and within the organism. And this is the system for higher organisms. Obviously, iron enters as ferric iron, it's reduced and held in the cell, and then heme, where's my cursor? Heme is absorbed in the intestine much more efficiently than iron itself. And so that's a particular problem we'll talk about with red meat. It's a way really to float iron into the duodenal cells. The exit of iron from the duodenal enterocytes is through an iron exporter with fairly limited expression, ferroportin. So iron is reconverted to ferric iron bound to transferrin, where it, of course, undergoes regulated entry into cells to be used for heme iron sulfur cluster synthesis. And any excess iron is stored as ferritin, which is a marker for iron stores in the organism. This transferrin-bound iron obviously also goes to the liver, where it interacts with transferrin receptor 2 and the HFE protein, and through a signal transduction pathway elaborates this very interesting peptide hepcidin, which then goes back and downregulates ferroportin, so competing a negative feedback loop for controlling iron. And there's also a bone marrow derived regulator of hepcidin, erythroferron, which also reports the bone marrow's need for iron. Within the cell, there's also this rapid and finely tuned response to too little or too much iron. As you all know, the mRNAs for the transferrin receptor, as well as ferritin, will bind iron regulatory proteins, IRPs. In the case of transferrin, when there's enough iron in the cell to bind to these IRPs, they pop off. The message is degraded, and so you don't make as much transferrin receptor, and therefore you don't bring any more iron into the cell. And conversely, at the 5' end, an IRP is bound to ferritin, blocking its translation until iron comes in the cell. And so when there is sufficient iron, you start making ferritin and sequester the iron. So literally, within seconds, we've evolved the system, which I think also speaks to the importance of its regulation. With the agrarian revolution about 10,000 years ago, however, iron deficiency became a bigger problem than iron excess. In fact, it still affects an estimated 2 billion people on the planet today. So more recent human evolution has produced mutations that have broken this tightly regulated system, creating conditions such as hereditary hemochromatosis, and also invalidation of the regulatory system I showed you before. All of the circled proteins can be mutated and cause iron overload, as well as iron deficiency in the case of ferroportin. Let's see. So the prototype disease illustrating the dangers of iron is the common form of hereditary hemochromatosis caused by most commonly one of two mutations in the HFE gene. And basically, when with mutations of the HFE gene that's not stably expressed at the cell surface, inhibiting this pathway that generates hepcidin. So without hepcidin there, the ferroportin channel is left over and the barn door is left open to allow iron to come into the cell with less regulation. There are other, obviously, regulators of hepcidin as well. So iron, you don't get totally iron overloaded. Many people can get into their fourth or fifth decades of life without evidence of any problems from carrying two mutations in the hemochromatosis gene, which is recessive. This disease was characterized in the 1800s as bronze diabetes, a syndrome of diabetes cirrhosis, and the skin coloration. And it's the bronzing both being due to the liver failure and jaundice, as well as the tissue deposition of iron. And it's a very common disorder. It's autosomal recessive, affecting about 1 in 200 Caucasians. So you can see the allele frequency is several percent. And it was probably positively selected in Northern Europe, where dietary iron content was low. I'm having trouble, yeah. So there was a question, though, early on about when the gene was cloned, that a lot more people were homozygous for the recessive mutations than had full form bronze diabetes. And so there was a question about whether it really was a disease or not. Ernst Beutler questioned that, largely based on the fact that the gene frequency wasn't really that different in 80-year-olds and in 8-month-olds. And so how could it be a serious disease? And so I had arrived in Utah and met Jim Kushner, who came to me and knew I worked on diabetes and told me he had something like 700 hemochromatosis homozygotes come through the GCRC, Clinical Research Center at the time. And he was working on heme porphyrin metabolism and said, I've got all these, but I've never really done anything with diabetes. Could you do anything with that? And by the way, I've got the mouse knockout also. And literally it was within days an RFA appeared, from NIDDK, prevalence and mechanism of diabetes and hemochromatosis. So I'm forever grateful to him, and he didn't know that was coming, forever grateful to him for getting me my only 0.1 percentile grant in my career, but so it was kind of a manna from heaven. It'd be hard to screw that up. So we were able to show using that it really was a disease, and we used, because of the large pedigrees there, affected probands and used their siblings as controls. These are all adults over the age of 45. And what you can see in this largely Northern European population, there, in fact, in the first cohort, there weren't any people with type 2 diabetes, but you can see in the hemochromatosis individuals on glucose tolerance testing, less than half had normal glucose tolerance, and about 25% had diabetes. And those are 95% confidence intervals, not SEs. So, so it did seem to be a health problem. And of course, he had also earlier shown that liver fibrosis exists even in medically unselected probands, sibling probands. And basically, we were able to show in the knockout mouse that the main problem in diabetes seemed to be lack of insulin production. This is actually islet area. We showed increased markers of apoptosis in beta cells stained red here by insulin. And so I thought the story was kind of going to be over in 6 months, but it turned out to be a lot more complicated than that. We were able to show that iron was causal in the problem of diabetes by bloodletting, which was, of course, one of the earliest medical procedures and still even practiced in the late 19th century. And we were able to show this, for example, in this small study where after phlebotomy, you can see the insulin area under the curve after glucose tolerance testing, approximately doubling uniformly in the individuals. And so they were able to rescue their insulin secretory capacity. There are other disorders with pathologic iron overload. Many of these are due to transfusions for anemia related to decreased red cell production or increased destruction. For example, beta thalassemia, sickle cell disease, and after bone marrow transplants. And like hemochromatosis, all of these are not only associated with diabetes, but also with cardiomyopathy, cirrhosis, hepatocellular carcinoma, and susceptibility to neurodegenerative diseases. So implying that iron plays a role in all of that. And here is just one of any number of studies showing the association of iron and thalassemia with average blood glucose on continuous glucose monitoring. And you can see a fairly good correlation there between ferritin levels and increased glycemia in that population. Now, obviously, you don't want to treat iron overload and anemia by phlebotomy, which would make the anemia worse, of course. So these situations, iron chelators are used, and we have both oral and IV, as you all know. And beginning in Europe, they were more aggressive, I think, than in the US, treating patients with thalassemia with chelating agents. And as a result, the prevalence of diabetes in thal is decreasing. Now, a similar situation exists in sickle cell disease, where iron overload is not only caused by transfusions, but also the intravascular hemolysis that's going on. Until recently, diabetes, though, wasn't really a problem because, sadly, patients with sickle disease were not living that long. But with better treatment of crises and chronic disease management, there are people now with sickle cell disease living into their 6th decade and beyond. So I'm working with a SCD expert at Illinois, Chicago, Victor Gordiuk. And we're seeing rates of diabetes in sicklers that parallel very closely the rates seen in the general population of African Americans, as shown here. And you can see in the very small n, though, it's even going off the graph here, the rate of diabetes is majority in the very few patients with male sickle cell disease in their 70s. I should also point out that people with sickle cell disease and diabetes are also disproportionately affected by higher rates, up to 2 to 3-fold higher, of hypertension, dyslipidemia, nephropathy, neuropathy, and stroke. So aspects of the so-called metabolic syndrome, as well as complications of diabetes. And as was the case in hemochromatosis, iron levels predict fasting glucose. In the first panel up here, fasting insulin in the second panel, and insulin resistance in the third panel. So you can see individuals, the higher the ferritin, the higher the insulin resistance. And you just breaking between ferritins of below and above 2,000, which is a kind of common goal for chelation therapy in sickle cell disease, although it's under-practiced, I still think. You can see that the proportion with impaired fasting glucose or diabetes is, again, a majority. Like patients with hemochromatosis, it turns out, though, patients with sickle cell disease tend to be lean. It's not totally clear why, but they are hypermetabolic, again, like patients with hemochromatosis. In our mouse models, we've shown that simply feeding a high iron diet can reproduce accelerated rates of fatty acid oxidation. So that's at least part of it. Although people with sickle disease are also, ill, frequently ill, and have multiple reasons for being thin, probably. Regardless, though, the lack of overweight is striking. 6.5% overweight or obese in this cohort with diabetes compared to 60% in a comparable non-sickle cell NHANES cohort being overweight or diabetic. So based on weight alone, you'd expect the prevalence of diabetes to be a tiny fraction of what is seen. And we think it's resulting from multiple hits of iron-induced insulin deficiency, like in hemochromatosis, on top of iron-induced insulin resistance, all laid on top of actually a unique genetic predisposition to diabetes in people with sickle cell. ApoB, for example, is a marker for diabetes risk, particularly in this population. So we're trying now to get a funded study to be more aggressive about iron chelation in this population, hoping to do to sickle cell disease what's been done to thalassemia. There's an interesting twist to the difference in hemochromatosis, though, versus SCD and thalassemia. Namely, the latter two are associated with insulin resistance, whereas patients with hemochromatosis are actually highly insulin sensitive. And this difference is explained by the fact that hemochromatosis is a low hepcidin state, whereas in most cases of SCD and thal, hepcidin is high because of the high iron. In hereditary hemochromatosis on the right, as you all know, no doubt, hepcidin, low hepcidin lets excess iron into the circulation, but paradoxically, cells that express a lot of the hepcidin-regulated ferroportin are iron underloaded in hemochromatosis. So you see less iron in the villus enterocytes and in cells that have a lot of ferroportin. The macrophage, for example, has to export iron to nurse developing red cells. And so the old before we knew the gene, the pathologic diagnosis was based on iron overload in hepatocytes, but actually paradoxical iron underload in the Kupffer cells. In dietary or high hepcidin states like sickle and thalassemia, though hepcidin is high and the iron is trapped within the macrophages and villus enterocytes. That has consequences. For example, we were able to show that iron downregulates adiponectin, which is a adipokine produced by adipocytes, and we have shown that it's responsible for much of the insulin resistance and dietary iron overload. But people with iron overload from hemochromatosis, on the other hand, have low intracellular adipocyte iron and therefore sky-high adiponectin, so they're very sensitive. And it's only when those individuals with hemochromatosis acquire insulin resistance, usually from obesity, that they develop diabetes, and then at higher than expected rates because they already have built-in insulin deficiency from that iron overload. So about 80% of obese hemochromatosis patients have diabetes. It turns out leptin, another hormone produced by adipocytes that regulates appetite and metabolism, is also downregulated by iron. These are actually associational data in a large human cohort we studied. But it's also true in mice where we've experimentally shown that the effect can be replicated simply by feeding diets with different iron content and in cultured cells. And the changes in leptin are reflected in the predicted changes in feeding behavior. So, and finally, the effect of iron on feeding is not seen in the OB mouse that lacks leptin. So we do think it's physiologically important in regulating appetite. And so as a result, we think the adipocyte really performs a special function, not only interpreting our macronutrient status, which we knew for the rest of the body, that is how many calories do we have stored, what can we do with that, but the status also of iron because of the reasons going back to the yeast story, the coupling of iron to metabolic regulation. So the adipocyte expresses a host of proteins normally seen only in iron responsive cells, including ferroportin and hepcidin. And it figures iron into the equation when it tells the brain via leptin whether you're starving or not, and when it tells peripheral tissues via adiponectin how much and what kind of fuel you ought to be burning. We put a lot of effort into understanding the mechanism by which iron regulates all these metabolic processes. I'm not going to show you everything today, but basically a lot of it is transcriptional. And what we really come up with is a who's who of transcription factors that regulate metabolism and the fasted fed tradition. So adiponectin regulation maps to FOXO1 sites, leptin regulation maps to cyclic AMP response element binding sites, Krebs sites, and in work I'm not showing today, iron regulation and circadian rhythm maps to PGC1 alpha. And so when we looked at cell autonomous stimulation of fatty acid oxidation that I mentioned earlier that occurs with high iron, AMPK inserts popped up. It's regulation in regulation by glycolysis. The hypoxia-inducible factors play a role. mTOR is regulated by iron. So we really feel kind of honored to be in this exalted gallery of factors, which are the really central regulators of metabolism. But it's also a little bit daunting because what could iron be doing to affect what is essentially the entire world of metabolism? And we reasoned it had to be some very proximal basic regulatory system. And because I worked on it earlier in my career and not any grander thinking than that, I went back to my earlier life when I worked on protein O-glycosylation. So this protein O-glycosylation, namely hooking UDP glucanac onto proteins, it's a little like Henny Youngman. It's had some trouble getting the respect I think it deserves. It's modification of serines and threonines by this N-acetylglucosamine. It's highly dynamic, but it's controlled by a single O-glycosyl transferase and a single O-glucanacase. It is not only analogous to phosphorylation cycling, shown on the right half of this slide, but in many cases, the precise same residues are being modified by either phosphate and O-glucanac in a yin-yang fashion. So one of the reasons that it has not been as widely studied as phosphorylation, it's a lot harder to work on because you either knock out the OGT or the OGA and it's embryonic lethal. The modified residues are these blobby hydrated bags that make it hard to make site-specific antibodies. And the residues also pop off in standard mass specs, so they're harder to map. And finally, if you mutate a serine, thinking you're affecting a phosphorylation site, well, you're also affecting the glycosylation site. So the response of a lot of investigators, I think, is just conveniently ignore it. But we showed many years ago that just sending extra substrate into these pathways could really uncannily reproduce type 2 diabetes phenotype tissue by tissue in fat, liver, muscle, and beta cell. O-glucanac is really nature's kind of perfect nutrient sensing system, and it is least in part substrate limited by UDP and acetylglucosamine, which contains all your major food groups, really. It's a high energy phosphate compound. The nitrogen comes from glutamine, It's rate limited by glucose flux into the cell here, and also the fatty acids contribute the acetyl group, and it is a it's a nucleotide as well, and uridine has been shown to be rate limiting for its production, glucose, and to some extent fatty acids as well. Nearly every transcription factor known can carry O-glucanac. And this is the way, for example, that Louis Ptacek, formerly at Utah, showed that nutrient status is imported to the circadian clock system. So our hypothesis was maybe that iron exerts its effects through this system. And we were able to show in our studies of regulation of leptin that iron increases Krebs phosphorylation, but it didn't seem to be activating any of the Krebs kinases. And this was really the initial link into O-glucanac. But it turned out it decreased both global O-glycosylation in proteins, as well specifically, as you can see in this lane, glycosylation. This is ferric ammonium citrate treated cells, glycosylation of immunoprecipitated Krebs without changing total Krebs. And this results in decreased occupancy of Krebs, this decreased glycosylation that mapped to the iron inhibition of leptin. And we were able to show this also in mice that were heterozygous for the O-glucanase, therefore expressed higher levels of glucanacylated Krebs. So anyway, getting back to the adipocyte and iron, when I arrived at Wake Forest, a very bright genomics guy, S.K. Das, came to me, and he had performed measures of insulin sensitivity by actually a very complicated assay, the frequently sampled IV glucose tolerance test, and did fat and muscle biopsies in a large cohort, 300 non-diabetic African Americans. So he came to me because he knew I worked on iron and asked me why many iron-related genes were among those whose expression was most strongly correlated with SI and body mass index. And this is expression in the adipocyte. One of the many interesting polymorphisms he uncovered related to insulin sensitivity was one of the most potent, namely in the transferrin gene. Now, overall, transferrin expression is well correlated with SI, and he had a polymorphism on the transferrin gene that explained most of this variance in transferrin expression. Now, remember, the tissue he's looking at now is adipocyte, adipose tissue, and everyone thought that the only organ that made a lot of transferrin was the liver, so what's going on in the adipocyte? Well, one first obvious possibility was that insulin resistance was driving the expression of transferrin as a response to insulin resistance, so it was just a trivial reporter of the insulin resistant state of these people. But that was not the case because in fact the polymorphism was driving the transferrin expression. So unless Lamarck was right, the transferrin levels are not the result, but actually the cause of the insulin resistance. Furthermore, the polymorphism did not affect serum transferrin, whose main source is the liver, and it did not affect transferrin expression in hepatomas cells or in other studies where we looked at liver-specific transferrin expression through, informatics searches of libraries. Most amazingly, though, knocking down transferrin in this last bullet in cultured adipocytes recapitulates most actually of the expression profile that was seen in the humans with the polymorphism. And this includes big players in metabolism, including decreased levels of the insulin regulated glucose transporter GLUT4, adiponectin, CPT1B, a major regulator of fatty acid oxidation rates, and uncoupling protein 2 as well. I mentioned earlier that fat cells express a lot of iron-specific genes, and transferrin is one of them. What we see here is taking pre-adipocytes and differentiating them in culture, undifferentiated in blue down through purple. And you can see the transferrin message is increasing 200-fold in adipocytes as they differentiate. So what is the fat cell doing with transferrin, and how does that affect insulin sensitivity if serum transferrin is not being affected? So we reason it must be doing something locally. It also turned out that humans with the polymorphism do have an iron-specific phenotype vis-a-vis insulin sensitivity. Remember, I told you that high iron decreases insulin sensitivity. So in his cohort of 300 that had the wild type transferrin or the major variant polymorphism in the transferrin promoter, those with ferritins greater than 100 have lower insulin sensitivity than those with ferritins less than 100. But if you look at the ones with the minor variant, they all have lower insulin sensitivity, and it's no longer responsive to iron. So this, I think, demonstrates a nice significant gene environmental interaction for diabetes risk. Furthermore, the variant also has functional consequences. SK knocked down transferrin in a cultured adipocyte line, and we looked at cellular respiration, mitochondrial respiration by seahorse. And in red, you can see the normal level in this maximal, in this situation, in this situation, maximal spare respiratory capacity. And that can actually be augmented by iron, adding iron to these cells. But in the cells with the transferrin knockdown, you can see less than half of the normal levels of respiration. And rather than going up with iron, they actually go further down with iron. Why this occurs, we don't know yet. We do know that the knockdown cells have lower intracellular iron levels, so you might think that adding more iron would rectify that. But one hypothesis that we're trying to work with now is that if you've messed up the normal regulated uptake system with all the chaperoning we talked about, you may also be messing with the normally well-orchestrated movement of iron within cells, in a sense unleashing maybe rogue or unchaperoned iron, although that's purely speculation at this point. So what could transferrin be doing locally in adipose tissue? And Alyssa Hasty at Vanderbilt has demonstrated paracrine trafficking of iron between macrophages and adipocytes within fat tissue as an important response of high-fat feeding. Basically, if you feed a mouse a high-fat diet, macrophages will spit out their iron, and it ends up in the fat cells. And of course, to get the iron into those cells efficiently, you're going to need local transferrin. And so we think this could be playing a role. And this movement of iron could allow browning of white fat, that is, turning white fat tissue from purely storage function to an actual metabolic function, which increases thermogenesis, a result of which would be increased insulin sensitivity and decreased weight gain, which is exactly what you see in the individuals with the polymorphism, without the polymorphism, I'm sorry. So can a case be made for dietary iron even within the broad range of normal causing diabetes? And this is important also because the average dietary intake of iron has been climbing steadily. This is a cheat y-axis. It's 10 up to 20, so this is not as big a fold, but it's about a doubling close to a doubling over the past 40, 50, 60 years. A lot of this was caused by iron supplementation of foods in the 40s to try to cure hookworm in the South. But much of the later could also be due to increased red meat consumption. And sure enough, there is a relation between serum ferritin and diabetes in normal, that is non-hemochromatosis humans. This is that normal range of ferritin, almost a 15-fold range of normal. And you can see across normal ferritins, if you extrapolate out, the relative risk of diabetes goes up 5, 6, 7-fold, which is comparable to the risk engendered by obesity, which is a pretty big deal. Now, when these data appeared, the common wisdom, beware that term, by the way, common wisdom, was that this must reflect the inflammatory nature of diabetes, because ferritin is also an acute phase reactant that increases with inflammatory stress. It turns out, however, that the inflammatory nature of obesity really is a fairly minor player. It contributes about 3% to overall serum ferritin. The rest is accounted for by dietary iron. And even in advanced liver disease, as one example, serum ferritin reports biopsy-proven hepatic iron stores, much better than disease severity. And in this graph, they actually went back and looked at the people at the right-hand end of the curve and said, are you inflamed or what? And they did not have increased levels of C-reactive protein or TNF-alpha and other inflammatory markers. Our cohorts have shown the same thing. But what you do find at this end of the spectrum are red meat eaters, basically. So in the walking well population, it's pretty clear to me anyway, it's still argued, by some, but that high ferritin is a really highly reliable marker of high tissue iron. And in most normal individuals, that indicates high dietary iron. So does it play a causal role in diabetes? And we've developed these mouse dietary models that we feed low, normal, and high iron diets. Low normal and high normal, 35 and 500 mg per kg, are basically different formulations of normal chow that you'll find. Low iron is able in unstressed situations to maintain normal hematocrits and normal growth in these mice. So they're not overtly iron deficient, but at the low end. And then high iron leads to a relative increase in liver iron that's comparable to what you see in normal human beings. So we think this is a pretty good representation of vegans up to heavy red meat eaters. And what you can see is that HOMA-B, which is a derived index of insulin secretory capacity, goes down with high iron in these diets, and insulin resistance goes up with high iron in these diets. And these, again, are the two hallmarks really of type 2 diabetes: insulin resistance and subsequent beta cell failure. The one exception is the beta cell at very low iron, and we think because of the reliance on glucose oxidation to trigger insulin secretion, and therefore the heavy reliance on mitochondrial metabolism, that even though these animals are growing normally, they may not have the levels of mitochondrial oxidation in the beta cell. But again, that is also not yet proven. So we performed a small pilot study while I was still in Utah in the GCRC on garden variety type 2 diabetes, decreasing iron in individuals from the upper half of normal to the lowest quartile of normal by phlebotomy. And that was just donating 3 or 4 units of blood on average. We collaborated with this native Brazilian Colorado, Mark Burgeon, New Mexico. And basically the decrease in serum ferritin was directly proportional to the increase in disposition index, which is the product of insulin secretion and insulin sensitivity. And it's actually the best predictor of all of these indices on future diabetes. So that little pilot earned us an R01, which I'm doing now with John Buse at University of North Carolina. So bloodletting for garden variety type 2 diabetes, and we're well underway in that study. So finally, just a quick word or two about iron and other chronic diseases. And it's worth mentioning that most of the diseases I'm going to be talking about are not only associated, like diabetes, epidemiologically with iron, but they're also associated with aging and overnutrition. So I talked about diabetes and transfusional iron overload. It turns out that iron overload in individuals, these are mainly people with thalassemia, is also correlated with markers of serum markers of liver damage. So remember that people with hemochromatosis, for example, also get cirrhosis, and serum ALT and AST are the two markers of that. So this is correlated not only with serum ferritin, but also with the number of units of blood donated, indicating again that ferritin is reporting the iron overload engendered by those transfusions. Oops, so sorry. So we did a study feeding mice a so-called fast food diet. So it's high in fat. They also high in saturated fat, about 50% fat, and also includes fructose in the water. So they're not only eating their Big Macs and fries, they're also drinking their big gulps sodas. And these mice do develop liver damage, shown here by ALT, but we've actually also shown bridging fibrosis, the classic pathologic hallmark of non-alcoholic steatohepatitis. And you can see that the animals on the fast food diet after 5 or 6 months have higher serum ALTs than the animals on the normal chow. But also, so there's a relation here between iron. So the mice in the open bars on the low iron diet are relatively protected from this developing ALT. And when you take the livers out and actually look at the mRNA levels for collagen 1 alpha, you can see, which is the essentially the fibrotic molecule in hepatic cirrhosis. You can see that a low iron diet essentially completely protects those animals from the fibrosis, ongoing fibrosis engendered by that diet. We also found gene expression analysis, RNA-seq, and found out that much of this seemed to be mediated by TGF-beta pathways, which is a well-known player in hepatic fibrosis. And we also found, interestingly enough, a series of inhibition of differentiation, these ID genes, which are upregulated, and these are also associated with hepatocellular carcinoma, which is a long-term complication of NASH, non-alcoholic fatty liver disease, and non-alcoholic steatosis. Many of them are actually driven by BMP6, which is also upregulated, of course, in the animals on the high iron. It turns out that ferritin in the cerebrospinal fluid is actually also one of the best predictors of cognitive decline in Alzheimer's disease. So you can see here, survival probability means survival without cognitive, mild cognitive impairment. or I'm sorry, moving from mild to severe cognitive impairment. And you can see that the higher quartiles of CSF ferritin are associated with progressively fewer or higher rates of becoming cognitively impaired. It turns out telomere length, which is a marker for several age-related pathologies, is also modulated by iron. So if you look at individuals with long telomeres and short telomeres, those with high iron tend to have higher rates of short telomeres, and people with lower irons have lower rates of short telomeres. And this was recently replicated in another large cohort, the NHANES cohort. So finally, just wanted to talk about something that's kind of fun and real quick, that our best diabetes drug that we use is metformin, and no one really knows how it works yet, surprisingly, despite its being used, or drugs in its class being used for over 50 years. And a paper appeared in 2018, which developed an assay shown in this lower box here for protein-protein homomerization. So basically, he tailed onto each individual protein in the yeast proteome, in this case, an inactive piece of dihydrofolate reductase. So when two proteins dimerize, in this case, you recreate an active dihydrofolate reductase. And first of all, I was surprised that the number of proteins that undergo regulated homomerization, monomerization, is very large. 20–30% of all of the proteome does this, which was kind of surprising. But you can mark this by becoming resistant to methotrexate, which is an inhibitor of folate metabolism. So cells with extra active dihydrofolate reductase will be methotrexate resistant. So basically, you can tickle the cells however you want to, and after tickling, say which proteins in my library show changes in homomerization, indicated by either increased or decreased methotrexate sensitivity. And so basically, you tickle the cells and then tally up the proteins that changed with that tickling, and you feed the names of those proteins into the magic pathway machine and say, what pathway is this? And their proof of concept was dumping rapamycin on the yeast, and you look at the pathway analysis, and bing, right to the top goes, of course, the mTOR pathway. And so what happened when they added metformin to cells was iron starvation response. And basically, they saw changes in augmented glycolytic metabolism that you see with iron starvation, also changes in mTOR signaling, DNA repair, lifespan, folate metabolism as well. So really, they suggested that interference with iron homeostasis may lie at the heart of how metformin does its job in being the best diabetes drug in the world. And we've looked at that in our mice on low, normal, and high iron diets. And if you just put them on the fast food diets, they become diabetic. So over this period of time, you can see their change in fasting glucose increased by this much. Both the high iron and the low iron mice. But if you put those mice on metformin, metformin actually ends up decreasing the glucose, as it does in diabetes, but only those of the low iron mice. If you look at the high iron mice, they're essentially untouched by metformin. So we think this iron starvation response may be crucial, again, indicating this really tight coupling of metabolism to diabetes. So in summary, too much iron is bad for you, but so is too little. And our overarching hypothesis really is that there's a range of optimal iron that's much narrower than this very broad range of normal iron, which is largely based on do you eat a lot of red meat or not. And right before actually giving this talk, I added this little lower panel. I got an email from the guys below that remind you that the epidemiologic studies suggest that eating 1 pound of red meat a week doubles your risk for diabetes, which is kind of a shocking number. These are the folks that did the work in my current lab, Shalini, Philip, Sandy, Alex, and Jin Fang. I've lost that slide, sorry. And of course, the people I've collaborated with, including really just remarkable collaborations at Utah. I was so happy, proud to have worked with Lynn Jordy, Joe Prakal, Dennis Wingy, Jim Kushner, John Phillips, and Kaplan as well, of course. So anyway, with that, I'll take questions, and I'm sorry I went a little longer than I thought. I had a little trouble flipping my slides here. So.
Well, thank you, Don, for that talk. We have a couple of questions. The first one is from Tom Gantz, and it asks, how does the different iron loading of islet cells in mice versus humans affect the usefulness of mouse models? Islets load in people, but not in mice with hemochromatosis. That's some work by Mitch Knudsen.
The islets, we found that the islets did load with iron. You know, we actually took, and we did that in cell culture as well. So I would disagree with that. It's a function of the dietary iron that you put them in. Again, you know, humans with hemochromatosis, you have to wait 40 or 50 years to see these demonstrations. But we showed that with regulation of the transferrin receptor with actually iron measurements in cells. So the cytosol of purified islets was high in iron in these cells. So I think, you know, I think you just have to be careful of your models. If you're on a, if you use that 35 milligram chow, for example, you're relatively protected from the effects of hemochromatosis.
Okay, in a second follow-up question, in epidemiologic studies, how can you disentangle red meat iron from red meat fat?
Well, you can't. The fact is, though, you can, to the degree that you can replicate the effects that we may attribute to high ferritin with simple iron feeding in the mouse models, and correlate that with the fact that ferritin correlates with red meat consumption, I think I think the conclusion has to be that iron is part of the story, but I agree that it's not the entire story, that of course red meat is going to be bad for you for all sorts of reasons. It's bad for the planet for all sorts of reasons. So, you know, environmental and animal welfare wise. So I don't mean to think that iron is the only factor here, and it's not the only factor causing diabetes. People with low iron, if they're heroic enough, can eat their way into diabetes despite low iron. I think it's a major and modifiable risk factor. The degree to which people got better with normal run-of-the-mill diabetes, however, simply by decreasing iron overload, indicates it's not a trivial factor.
Right, thank you. Another question, do we know if serum ferritin in the brain is high in iron, as serum ferritin is typically thought to be low in iron?
Serum ferritin in the brain?
Yeah.
Oh, well, it's CSF ferritin. And I think it's still APO. I'm not, you know, it's not been, to my knowledge, maybe they haven't really looked at CSF ferritin to know that it's apoferritin, but the presumption is that it's going to be reporting it in the glial cells, you know, analogously to the way it reports iron iron stores in macrophages. But not, you know, the brain trafficking of iron is still, there's a lot to be learned there. You know, it has to take a transferrin-dependent pathway to get across the blood-brain barrier. Once it's in the brain, again, there's going to be hepcidin dependence there of egress from the glial cells. So, you know, I think you're going to have to really look at individual cell iron, and we actually have just collaborating with someone who's just gotten a piece of equipment that will allow us to do that, because we're looking at iron in degenerative disease, not only Alzheimer's, but also Parkinson's. Basically, if you take any kind of population with degenerative neurologic disease, like I said, Parkinson's, ALS even, that population is overloaded with both heterozygotes and homozygotes for hemochromatosis mutations. So.
Okay, we have two more questions. One is, what is the impact of iron chelator in iron overload induced diabetes or metabolic syndrome?
There's been a small study to use actually chelation. We did it in our mouse model and it worked fine. The fact is that if you're a normal individual without an anemia or without you know, say thalassemia, it's just a lot easier to do the phlebotomy to decrease the iron. So, you know, in our human trial, we, you know, it was so much safer that we did chelators. With many people with advanced diabetes, of course, diabetes and renal disease, they do have the anemia of chronic disease, and it may be important to test chelators in them. But it's really not, you know, not known yet. The best evidence would be in the thalassemias, where the iron reduction is done by chelation, and they both become more insulin sensitive and increase insulin production and have lower rates of diabetes.
Okay, and the final question, you mentioned a comment about a normal narrower range of iron in terms of ferritin. Is there a recommendation what that normal narrower range would be?
Well, no. You know, if you look at one of the European studies of phlebotomy, it's a fairly small study, but they did graded phlebotomy, and it appears that there may be, remember, very low iron doesn't support insulin production very well. And so there's an inflection of the curve around a ferritin of 50. So, you know, the minimum curve of that diabetes risk curve is around there, but that could be plus or minus a factor of 2 easily.
Okay, great. Thank you so much, Don, for the talk today, and thank you all for your attendance. That's it for the summer seminar series, and we'll begin again in the fall, starting, I believe, September 15th with Dr. Tom Gantz, who will speak to us, and I hope to see you all joining again. And you can unshare your mutes or undo your mutes and talk for a sec if anybody else wants to talk on this.
Thank you for having me.
Yeah, thanks, Don.
I just wanted to say hi, Don. It's Judy Simcox.
Oh, hey.
Hey, Judy. How's Wisconsin?
It's good. It's full of cheese and
okay. Awesome.
Yeah, and some good science.
Good. Glad to hear it. Okay. So I'll probably, I'll let you guys chat another time. I'm going to end the meeting. And thanks again, Don, very much. So nice to see you. Nice to see you, Judy, and everybody else. Yeah. Best wishes to all.
My pleasure. Thank you.